Hi and Assalamualaikum. My name is AISYAH SOFEA binti AZMIR, and today me and my groupmates are going to present to you about cell theory. But first, what is cell? Cell is the smallest basic structural and functioning unit that can live on its own, and all living organisms are made out of cells. They may exist as independent units of life, as in monads, or may form colonies or tissues, as in higher plants and animals. As you can see in this picture, a cell will create colonies of tissues, and then the tissues will create organ, and an organism was created. There are two types of cell, which is eukaryotes and prokaryotes. Alyna will brief to you about eukaryotes, while Shakirah will present to you about prokaryotes. But first, we need to know how the cell theory was born.
Hi and Assalamualaikum. Now I am going to tell you about who started to come out about the cell theory. These are the scientists who started to discover the cells. In 1665, Robert Hooke, who first started to discover cells, made his own book named Micrographia, which he explains and describes the cells in the book. He wrote over 60 observations of various objects under his compound microscope. One of the observations was from very thin slices of bottle stopper, or known as cork. He found the cork looked as if it was made from tiny pores, which he named it as cells. This came from the Latin word cella, meaning small room. However, Hooke did not find the very detail about the cell, the real function and structure, since he owned a low magnification of microscope during that time. Next, Anton van Leeuwenhoek, another scientist who has seen the cells soon after Robert Hooke did, by improving the lenses in microscope that could magnify objects almost 270 times. Under his microscope, Leeuwenhoek discovered motile objects. Over the time, he noted many papers which observed many specific forms of microorganisms. He called these as animalcules, which included protozoa and other unicellular organisms such as bacteria. He also found for the first time the sperm cells in animals and humans. Then a German scientist named Theodor Schwann, with the help of Matthias Schleiden, make research on cells of animals and plants, respectively. They identified key differences between the two types of cells and came out with the idea that cells are the fundamental units of both plants and animals. Unfortunately, they misunderstood about how the cells grow. One stated that cells seeded, while the other one believed that animal cells crystallized from material between other cells. But then, the problem was solved and identified by Rudolf Virchow in 1855, who declared that all cells are generated by existing cells. Over the years, scientists and biologists began to further study about genetics, chromosomes, and DNA. This had so much impact on science than Robert Hooke could have ever imagined in 1665.
Next, I'm going to present to you about the principles of cell theory. The first principle of cell theory is all organisms are composed of one or more cells. A parasitism is a unicellular, an alpaca and a bonsai is a multicellular, as it was created from more cells, while a parasitism is created from one cell only. The second principle of cell theory is the cell is the basic unit of structure and organization in organism. As you can see in this picture, a cell will divide it into more cells. It is called tissues. An organ was created from tissues, and an organ system will be created. An organism will be created if we have a perfect organ systems. And last but not least, the third principle of cell theory is cells arise from pre-existing cells. It means that the cells go through mitosis as it has identical daughter cells like the parent cell.
My name is Alyna Shakirah and I'm gonna explain about eukaryotes. There are 4 types of eukaryotic cells, which is animal cells, plant cells, fungi, and protists. Which one is larger, eukaryotes or prokaryotes? The answer is eukaryotes. It is because eukaryotic cells need to carry out a metabolic function that prokaryotic cells doesn't even do. So, eukaryotes. Eukaryotes are organism cells that contain a nucleus and a membrane-bound organelles. Eukaryotes may be single cells or multicellular cells. Eukaryotes different than prokaryotes because of the presence of cytoplasm in the region between the plasma membrane and nucleus. That's for eukaryotes. Meanwhile, for prokaryotes, the cytoplasm is the region inbound with the plasma membrane. So, types of organelles that consist in eukaryotes. First of all, we have nucleus, where the cell's genetic information are stored. It's organized in long molecules called chromosomes, and the nucleus is also the first place where the early stage of reproduction occurs, which is mitosis. Next one, we have mitochondria. Mitochondria generates the most of the chemical energy needed to power the biochemical reaction that occur in the eukaryotic cells. Chemical energy produced by the mitochondria is stored in a small molecule called adenosine triphosphate, or it's known as ATP. Next one, we have endoplasmic reticulum. The endoplasmic reticulum serves important function particularly in synthesizing, folding, modification, and transport of proteins and lipids. There are 2 types of endoplasmic reticulum, which is rough endoplasmic reticulum and smooth endoplasmic reticulum. So, next one we have Golgi apparatus. Golgi apparatus is an organelle that moves molecules from endoplasmic reticulum to their final destination. The organelle also modifies products of the endoplasmic reticulum to their final form. Last but not least, we have chloroplast. Chloroplast only exists in plant cell. It converts light energy into chemical energy, so it is the main organelle for photosynthesis process to occur. They harvest light energy to make ATP, sugars, and oxygen.
And now I will explain about prokaryotes. Prokaryote is one type of cells and divided into 2 types of kingdom, which are kingdom bacteria and kingdom archaea. Prokaryotic cells have such characteristics which are more simple cell, tiny in size, no nucleus as the DNA in unbound region called nucleoid, and no membrane-bound organelle. Parts of prokaryotic cells and its function. Capsule, a thick layer made of polysaccharide, function in protecting cells from drying out and help them attach to the surface. Cell wall, rigid and made from peptidoglycan, it provides structural support and maintains shape of cell. Plasma membrane is semi-permeable membrane for regulation of substance in and out of a cell. Cytoplasm is site of metabolism reaction in cell. Nuclear DNA is the region where the DNA is located. Plasmid DNA is sites of transfer between bacteria assumed as horizontal gene transfer. Ribosome is sites of protein synthesis. Pillars have 2 functions. The first function is attachment of pili, helping the bacteria adhere to the surface. And the second function is the sex pili for bacterial conjugation as they swap the DNA with one another. Flagellum for locomotion of cells in order to move around. And that's all for prokaryotic cell.